So welcome to everyone. I guess that we can start. It's 2 now, it's 2 p.m. So I'm Marilaure Grégoire and I'm co-chairing the Global Ocean Oxygen Network with Andreas Oschlies. And today I have the great honor to be the moderator of the 4th GO2NE webinar on ocean deoxygenation. So today we'll be talking about hypoxia, and we will have 2 great speakers with Wenfei and Daniel Conley. It will be organized as follows. So first we will have a presentation by Wenfei of 20 minutes, and then afterwards you will have the possibility to ask questions in the question box, please. So use the chat only for other purposes, but for the question, use the question box. For your information, the talks, the session will be recorded and will be available in a few weeks. So first, we will start now. So we'll start with Wenfei. Wenfei obtained a PhD about 1 year ago from the Maryland University, from the Center for Environmental Science. And she did a PhD on the impacts of climate and nutrient discharge on the Chesapeake Bay dead zone. She obtained a presidential fellowship to perform this PhD, and now she is working at the NOAA Climate Program Office. So, Wenfei, the floor is yours.
Sorry, it stopped for some reason.
Wenfei, can you share your screen?
Yes? Yeah. Okay. Cool. Now it's good. It's just stopped. Yeah.
No problem.
We don't see it yet. No, not yet. Wait a second then.
Okay, it's coming.
No, thank you.
Can you see it now?
Yes, we can see it, but it's good if you go to full screen, I guess. It's better.
Yeah. Okay, I'll go. Okay, that looks good. Okay.
Is it the full?
Yeah, it's full screen on my side now.
Okay.
It looks good on your side too.
Perfect, Wenfei.
Okay.
Thank you.
Okay, I'll start. Thank you. I don't know. Sorry. It's my just the app, the software got stuck my side for some reason. I cannot. Okay, perfect. Sorry for the technical issues. Being a little trouble. Thank you for the great introduction, and thank you all for being here today for the presentation. I'm glad to have this opportunity to share my recent research work with my advisor Ming Li and Jeremy Testa at University of Maryland Center for Environmental Science on the topic about the impact of nutrient management and the climate change on the long-term change of Chesapeake Bay hypoxia. This work was supported by the United States NSF and NOAA. Oxygen concentration in both the open ocean and the coastal waters have been declining since 1950s. Unlike the open ocean, where the deoxygenation is primarily caused by the global warming, in estuaries and coastal regions, the hypoxic spot generally occurred at estuaries such as Chesapeake Bay, Long Island Sound in the United States, and the river-dominated shelves like Northern Gulf of Mexico and East China Sea, and also upwelling systems like California upwelling system. the incidence of hypoxia has increased almost 34-fold in the United States since 1960s, with more than 300 systems recently experiencing hypoxia. The oxygen declines in these systems have been mostly caused by the increased loadings of nutrients from their watersheds. The mechanism of eutrophication-induced hypoxia is very simple, that with excessive nutrient input into the estuaries, it promotes algae overgrowth in the surface water. As dead algae decomposed by the bacteria, oxygen is consumed in that process. Well, the oxygen replenishment from the upper layer turned out to be not sufficient. The hypoxia occurs in the bottom water. And this recurring low oxygen condition would threaten the ecological health of the coastal waters, leading to like fish kills, the declining water clarity, and harmful algal blooms, acidification, and habitat degradation, and ultimately reduce the ecological service that it can provide. Being aware that the excessive nutrient pollution as the main source of coastal hypoxia and ecosystem degradation, controls on the watershed pollution have been implemented since 1980s in some of the hypoxic waters, however, with different management efforts. As expected, with substantial nutrient reduction actions, they're mostly, we can see the major improvement of oxygen condition in the systems like Tampa Bay, Northwest Black Sea, not like Northern Adriatic Sea. With modest nutrient reduction, we can mostly see the local recovery of water quality, and there's a little sign of even like no sign of hypoxia mitigation in the cases like Chesapeake Bay and the Baltic Sea. Not surprisingly, if there's no nutrient reduction, there's going to be no decline in the extent and the duration of hypoxia. Such case is like in the northern Gulf of Mexico. Meanwhile, the climate change exacerbates the oxygen decline in coastal systems through the similar mechanisms as those in the open ocean, as summarized in the chart by the Attieri in 2015 paper, among which the altered precipitation pattern and sea level rise are the most prominent climate drivers. Studies in two of the largest coastal dead zones in Baltic Sea and northern Gulf of Mexico have suggested the likely increase of hypoxia with future climate change climate model projection, with the cases of various nutrient load scenarios. So this indicate a potential computation between those two forcings, the climate drivers and nutrient the drivers. Because these two large systems are unique in the spatial dynamics, so estuaries with shorter residence time and faster response to the nutrient load will be the target of this research. So here we set our sights on the past decades when the watershed nutrient reduction began, and aim to answer those two questions. What are the roles of climate drivers in the long-term change of oxygen level? And do they compete with nutrient reduction to elevate the hypoxia? In this study, I take Chesapeake Bay as my study area for those two questions, because the Chesapeake Bay has a relatively long history of eutrophication and hypoxia. Also, it has a kind of relatively plenty monitor data. And Chesapeake Bay is the largest estuary in the United States on the East Coast. Every summer, the hypoxic water stretch along the central deep channel, which is about 20 to 30 meters. It can take up to 1/3 of the bay water below 10-meter depths. In Chesapeake Bay, the hypoxic volume increased synchronously with the increasing input of nitrogen since 1950s. However, with a series of Chesapeake Bay agreement to reduce nutrient pollution from 1983, and as well as the most recent Chesapeake Bay total maximum daily load (TMDL) in 2010. There is a modest reduction of the nutrient input at the largest tributary Susquehanna River of Chesapeake Bay, with about 15% decline of nitrate and about 25% decline of phosphate. But during this during this period of time, there's only the water quality restoration result turned out that only local recovery of seagrass bed, and there's no there hasn't been any publications showing there's any decrease in the summer hypoxic volume maximum. Although there might be there's some studies shows there's some change in the timing of hypoxia. Also, the Chesapeake Bay has suffering from regional climate change for decades. The surface water temperature has been increased by about 1.5 degrees, and the sea level rise. Also, there's a prominent sea level rise on the ocean boundary. In this study, we discerned the impact of climate and nutrient drivers with numerical simulations under different scenarios. So we used the offline coupled physical and biogeochemical model ROMS RCA to implement those simulations, focusing on two middle bay regions where mostly the hypoxia occurs. In the biochemical model, if you're interested, it simulates the cycling of carbon, oxygen, nitrogen, phosphorus, silicon, sulfur in both the water column and the sediment. Most importantly, for this study, we designed 4 simulations model scenarios to answer the questions we asked at the previous slides. So first is a handcast simulation from 1985 to 2016. We call it base run, and also with other 3 model scenarios. The first one is the trend sea level rise scenario, which we just extract the sea level rise trend from the handcast simulation. So this is a simulation with at low sea level state. And then second is the trend temperature scenario, which similarly we just remove the warming signal in the model for things, and it kind of represents the cold climate over the same time period. and the trend nutrient reduction scenario, which is just we assume there's no reduction occurs, so the concentration of the nitrate and phosphate concentration maintained at high level. So keep this for model scenario in mind. Yeah, these 4 figures just shows how the climate drivers looks like and the nutrient load looks like on the model boundary. So it's very obvious there's a sea level rise trend in the open ocean boundary, and also the increasing trend in the surface air temperature in the atmosphere ocean boundary. The solid black lines in the 2 bottom panel, just that is a like observed like historical nutrient input, but the red dashed line is the like nutrient concentrations when there's no reduction. So we just add back the historical reduction, and assuming there's no reduction happened. And so those are the 4 things we're going to use for the scenario. Since Chesapeake Bay is a relatively shallow and productive estuary, the bottom oxygen in the central channel has prominent seasonal cycles, and also with certain interannual variabilities. So in order to extract the long-term trend, we use a general additive model to separate the long-term change from the large seasonal and interannual variations. Because with GAM, we could construct the nonlinear smooth terms for the trend at different time scales and only keep the long-term trend information that we need. Here's a monthly averaged ROMS-RCA output of surface bottom oxygen averaged by region is the input for this statistical model. First of all, we examine the climate change signal reviewed by the HANCA simulation in comparison with the limited observation at certain stations in the bay. So both the model can both capture well the sea level rise rate as well as the temperature increase in the bay, which is about 15 centimeters over the 32 years and 1.5 degrees over the period of time. Both the model and the observation show similar trend. So the constructed GAM model, first it proved that it can well reproduce the variation of both surface and bottom oxygen concentration, which shows in the upper 2 panel. If we look at the long term, the smooth term of the long term oxygen trend, which is the red solid line showing at the middle of the surface and bottom oxygen in the middle bay region of the bay. So the long-term trend is very clear. The long-term trend is very small compared to the variance of the oxygen. So that in turn verify that we need the certain of special treatment of the trend analysis. And the bottom oxygen declined about by 0.3 milligram per liter over the 32 years. However, if we look closely into the seasonal trend of bottom oxygen, not every season reviewed the significant declining trend. So there's gonna be a faster bottom oxygen decline in winter and spring, which is statistically significant. Well, it turned to be smaller decrease in summer and even slightly increase in the fall. So it could explain why both model and observation did not show any significant trend in the hypoxic volume, because the trend in summer is relatively subtle. Putting together the smooth terms for the long-term trend for the 4 different scenarios together, for both surface and bottom oxygen, it is very intuitive to tell that like most of the scenarios, in addition, except the so in most of the scenarios there shows a long-term declining trend in the bottom in the oxygen concentration, and only in the scenario with detrended warming signals, there's gonna be there's no declining trend or even slightly increased trend in the bottom oxygen. So that tells us the temperature increase as the main driver for the long-term oxygen decline. Comparing the scenarios with the base run in the most recent decades, when the differences in the forcing are most prominent, like the panel on the right showed us, it tells both the sea level rise and warming generate negative effect on the oxygen level, and the nutrient reduction induced restoration is all overwhelmed, offset by the climate change signal. Further, also we check how these scenarios, how the timing of the hypoxia change response to the different scenario runs. So this just shows the difference. So the panel on the right just shows the difference of the date of hypoxia on site between the scenario runs and the base runs. So it's clearly show that the warming and the nutrient reduction has counteract on the timing of the hypoxia initiation. So the warming would shift the hypoxia early by about 6 to 10 days, while the nutrient reduction will postpone it. However, both the warming and the nutrient reduction gonna drive an earlier termination, so they kind of agree in this case in the timing of the hypoxia termination. So there's a brief conclusion of this research is that in the Chesapeake Bay study, the warming dominates the long-term oxygen decline, especially in the winter to spring time period. And it definitely offsets the benefit of nutrient reduction to mitigate the summer hypoxia in the past 3 decades. And therefore, more aggressive actions will be required to counter the anthropogenic eutrophication under the climate change background. And so this research is still, I have still been working on this for some further study based on this research to investigate the combined effect of changing climate and the nutrient load scenarios with ensemble future climate projections. And I will try to evaluate the sensitivities and the certainties in the model projections, considering both the climate projection and also the nutrient scenarios. And the last thing is a rough source, but it's an interesting idea that in the MyChem's 2009 review paper, there's a hypothesis about the hypoxia recovery trajectory, with and the regime shift in the biogeochemical cycle with the change of the nutrient load. So I think the numerical model is a good tool to look into this topic and trying to answer some of the questions hasn't been solved yet. So that's my talk today. Thank you very much.
Thank you very much, Wenfei. It was very, very interesting, very comprehensive, and it showed all the potentiality of using modeling. Thank you so much. So now we'll go to the questions. So I invite all of you to put the question in the chat. So I have here a question from Andreas Ochlis, and the question is about what if Andreas is asking you if the increase in winter temperature or summer temperature, it's the main. What is the you say that the increase in temperature was very important. What is the main factor? Increase in summer, increase in winter, or increase in spring?
You mean the seasonal variation of the temperature increase, right?
Yeah, is it the annual mean variation that is important, or is it really the increase in some season that's really drive the systems?
For the temperature increase, I haven't looked at it by seasonal, but I got the impression that the temperature increase in the Chesapeake Bay is there's not too many seasonal variations, so they all increased significantly. I think what most important thing is that during the winter with the same amount of increase in the temperature, it's just the solubility of the oxygen is mostly changed. It's just being changed more largely than in the summer because of the property of the oxygen in the water. Also, like in the winter, that's when the fresh water discharge peaks, winter to springtime. So I think the fresh water also affect the change of solubility. So I think, yeah, I think the oxygen solubility is kind of is the main cause for the temperature impact. Yeah.
So I have here another question. What is the specific impact of the sea level rise on oxygen?
Sorry, can you repeat that again?
What is the specific impact of the sea level rise on oxygen?
Okay, I think the sea level rise on oxygen. So because Chesapeake Bay, I forgot to mention, Chesapeake Bay is a partially stratified estuaries. So with the sea level rise, it can definitely change the circulation pattern in the Chesapeake Bay and as well as mixing. So the stratification will. I have another paper talking about this. So the stratification mostly increased in the bay with sea level rise. So that definitely would lead to more hypoxia. Yeah, I think that's the stratification. Also, the mixing and the vertical advection. So the physical, the dynamics of the transport of the oxygen.
Yeah, so in your perspective, you mentioned that you would like to run ensemble simulation, which is a very good idea. And so if you have to define your ensemble, what would be the forcing or the parameters that you will change? And there is a question here about the importance of the wind patterns. So do you think that the forcing by the wind could change a lot your circulation in the bay and then change the dynamics of hypoxia? What would be the main field that you will perturb first?
Yeah, that's a great question. First of all, I'll answer the question of the wind pattern. So the wind is a tricky part in the climate projection. So because for the temperature, we kind of have a more consistent impression of it's going to be warming in the future. But for the wind pattern, like the change of the wind speed and the change of the wind direction is on the regional scale. I think for now it hasn't been well resolved. So there's not too much agreement among the models, and as well as the precipitation. So these two are kind of the most tricky part. The precipitation, like if you check the regional climate model, downscaling models, it's gonna be mostly spread for the precipitation as well as the river discharge. So And yeah, temperature is like the directions all positive, it's just the matter of degree. So yeah, if I'm gonna choose the parameters, I will leave the wind aside and choose the temperature and river discharge, because it's estuary, so river discharge is very important. So
okay, so do you think that there is another question here, but just briefly, do you think that the river discharge that you are imposing is really forcing your system? Do you think that your conclusion can change if you had other conditions at the river? Is it very sensitive? Is it really forcing that affect your model simulation?
yeah,
Okay.
that's another good question. So I think river discharge definitely play an important role, because river input is a driver for the hypoxia in the Chesapeake Bay. But besides the large uncertainties of the future projection of river discharge, I think I feel it's just I haven't done the research yet, but it's just my initial impression. I feel it's the temperature should still would still dominate the climate changes, while the river discharge, you have to, I think in that case you have to combine with what nutrient load scenarios you're gonna take. So the combination can lead to a huge change of the nutrient load into the bay, which I think it would probably have a large influence.
Yeah, and so you mentioned the temperature is very important, so we understand that very clearly. And so there is another question here. So do you think, what is for you the most important impact of temperature? Is it through solubility changes, or is it through degradation and respiration? Did you try to investigate the model?
Yeah, that's a great question. Yeah, I have another paper in 2019. I just use climate model projections to study the future climate change impact on the hypoxia in Chesapeake Bay. So in that research, I found, I think the solubility account for like about half of the temperature impact, especially again in the winter and the springtime. But as you said, the temperature induced change in the ecosystem, like a metabolism rate, mostly occurs. There's also like a 20–30%, but it's has very seasonal differences as the temperature. So it depends on which like season you are focusing on, and yeah.
Okay, thank you very much, Wenfei. Thank you very much for this interesting talk. Thank you. So now we'll move now to our second speaker. So it's Dan Conley. I don't think that I really need to introduce Dan, so everybody knows him. But anyway, I will try with just a few sentences. So Dan is professor at the Lund University in Sweden. He did a lot of work on the Baltic Sea and understanding the hypoxia dynamics, and then recently got advanced grant from the ERC for investigating the silicate cycling and the impact of diatoms of this cycling. But today, Dan will speak about the Baltic Sea, and we'll speak about the dynamics of hypoxia, many shapes and many sizes. So Dan, the floor is yours.
Thank you very much. It's certainly a pleasure to be here to talk about the Baltic Sea. It's one of my favorite topics. The work that I'm talking about has been done over a period of 20 years. A lot of the work has been together with Jakob Carstensen. So he's also a co-author on this talk that we're giving today. And I also want to thank our supporters. We've actually have had very generous support from many groups in the Baltic Sea that are interested in hypoxia. Okay. So what I'm going to talk about today is, where does hypoxia occur in the Baltic Sea? Is it in the open waters? Is it in the coastal zone? Is it in the northern part, the southern part? I'll talk about, when does hypoxia occur? Is it historical? Has it always been there? Is it perennial like it is today, which means it's there every year throughout the year? Is it annual, something that only happens during the summer? Or is it episodic, something that comes and goes? And then the important thing is this leads to the mechanisms that create hypoxia. And then I'll leave a few words about perspectives for the future. So one of the things you should keep in mind, this is a picture of the Baltic Sea. It's in northwestern Europe, but the Baltic Sea is naturally prone to hypoxia, and it has to do with how it's set up. One of the things you can see is that saltwater intrusions come in only through a very narrow opening through the Danish Straits. And the amazing thing about that is that there are several sills there through the Danish Straits that range from 8 meters to 18 meters. So it really restricts the amount of water that comes in. So you need very special conditions for salt water to intrude to the Baltic, and they tend to be climate driven. You need strong winds from the east to drive water out for several weeks, and then you would need another system to come in that is coming from the southwest that will push salt water into the estuary once again. And it's something that doesn't happen every year. It probably happens on average a frequency of once every 10 years. And then you have the Baltic proper, and that's where most of the hypoxia occurs, is in this enclosed region. If you go up to the Bothnian Sea and the Bothnian Bay, they're much less productive and much more fresh than the Baltic proper. The Baltic proper has salinities between 6 and 8, whereas the Bothnian Sea is between 4 and 6, depending on where you are. It is about 1,500 kilometers from the very southern part up to the northern part of the Baltic Sea. So it's a very large ecosystem. One of the things that's well known about the Baltic Sea and is well known from other places is that you've had huge increases in nutrient loading, especially during the last 120 years. And you can see this in the figure. Most of the largest increases in nutrients for nitrogen and phosphorus came with the use of fertilizers in the 1950s, but also sewage inputs. And then it peaked between 1970 and 1990. And during that time, they made a number of changes in nutrient usage in the watershed, both through agriculture, but also through wastewater treatment plants. And one of the things you can see is both phosphorus and nitrogen have decreased over the last 20 or 30 years. And these reductions are needed to reduce the amount of hypoxia in the system. And then you also have this solid blue line and solid red line that gives the targets where they want to reduce the nutrient levels to. So in 2002, we published this paper, and it was the first use of using hypoxic areas. And we took this concept from how they look at hypoxia in Chesapeake Bay, is how many square kilometers does it cover? And unfortunately, the Baltic Sea is the largest hypoxic area in the world, going as high as 60 and 70,000 square kilometers. But one of the interesting things when we put this together, I should note that the squares are the winter values and the dots are the summer values. And it was integrated over the entire Baltic proper. And one of the things you see is that it doesn't necessarily follow the nutrient inputs. The highest oxygen actually occurs during this thing called a stagnation period, when there were few saltwater inputs into the Baltic. So this period really between 1980 and 1993, before a new saltwater input came in, you can see it has much lower amounts of hypoxic area. And the importance of saltwater is that it increases stratification. And when it increases stratification, you have more oxygen, confined oxygen consumption that leads to more hypoxia. But this was done relatively crudely, given the computer resources and the techniques we had at that time. And so the question is what happened prior to 1970? And it turns out that there's data available for the Baltic Sea that goes all the way back to 1888. The Winkler technique was invented in 1888, and it applied to the Baltic Sea soon after. And so we used statistical-based models to look at changes in apparent oxygen utilization and then extrapolate that into how much is consumed throughout different periods. And we can see this rapid increase in hypoxic area already beginning in the 1900s and going to what we see today, which is 60 to 70,000 square kilometers. And it's a 10-fold expansion in hypoxic area. And we know that there are different influences on hypoxia. And one of them, of course, are nutrient inputs. And so this rapid increase in the 50 probably had a lot to do with fertilizer use and wastewater treatment inputs. And then you also had the saltwater inflows that also helped to mitigate. At first, it mitigates the hypoxia, but then when saltwater comes into the Baltic, it stratifies it stronger, and you have more, less oxygen being able to make it down to the deeper waters, less mixing, and so you have more hypoxia. And this is also interesting to note that the residence time of the Baltic proper is on the order of 30 years. So we're really suffering from the sins of the past. The largest inputs of nutrients occurred between 1970 and 1990. And so that also means that many of those nutrients are still cycling within the Baltic today. And if you also look at the bottom figures, you have three different periods: 1906, 1955, and I can't see on my screen here. Anyway, one of the things you see is that the area of hypoxia has increased tremendously. It's now on the order of 65 to 70,000 square kilometers. The black is anoxia and the red is hypoxia. So all the deepest areas have problems with hypoxia, and it's actually anoxia in many of the situations. So I think one of the questions we had was the Baltic Sea always hypoxic? So we did a study looking at sediment cores, and we used laminated sediments as indicators of hypoxia. And that's because when there's no oxygen, benthic invertebrates cannot live in the sediments, and they don't mix the sediments. And so the layers are clearly formed. And you can see there are actually three periods of hypoxia over the last 8,000 years. And that's the time period where the Baltic Sea, the modern Baltic Sea, has formed. And the first period was during the Holocene thermal maximum. And we had very warm temperatures during this period of time. And it was also lots of saltwater input. The opening through the Danish Straits was larger, but then as you had isostatic rebound and the land arose after the glaciers left, it constricted the amount of salt water that could go in. And then there was a period of more than 2,000 years where there was no hypoxia. And then we had the medieval climate anomaly. Many people know it as the medieval warm period, where we also have hypoxia. And this is probably largely related to increases in temperature, as also shown in the previous talk as well. And then you have the most recent increases in hypoxia that occurred the strongest since the 1950s, but going back to the 1900s. And it's due to increases in nutrient inputs into the Baltic Sea. And so what it meant is that hypoxia hasn't been in the Baltic Sea throughout its entire history. There's been periods of time where there has not been hypoxia, which gives us hope for maybe bringing the system to a state where we have at least less hypoxia than we have today. So what do we know about coastal hypoxia? One of the interesting things was that the data for the open sea has been collected for many, many decades. There were agreements already in the 1950s and 60s that all the countries around the Baltic Sea would share their data. And so even today, everything is online. Every time a research cruise goes out and collects data, they deposit it at ICES, the International Council for the Exploration of the Seas. And so all the data is there for the open waters. But coastal hypoxia, the data is very much held by the countries because it's within their territorial waters. And so in 2011, we went and asked a number of different countries for their coastal data and analyzed it. And we found a number of important things. We found that hypoxia is actually widespread throughout the Baltic Sea. One of the interesting things, if you look along the coast of Germany and Poland and Latvia and Estonia and Lithuania, there's no red dots, which are hypoxia, and there's no black dots, which are anoxia. And it's because these are very open coastlines, they're sandy beaches, and you have mixing. And as opposed to if you look at Finland along its southern coast and the eastern coast of Sweden, and a lot of the enclosed areas in Denmark and Germany, and also along the west coast of Sweden, and especially in the archipelagos, the two offsets there, you can see tremendous numbers of red and black, meaning that hypoxia is widespread in these areas. And it's because they have they're enclosed, and the mixing processes are very important in determining whether you have hypoxia or not. And it's also close to the sources of the nutrients. So we found that 215 of the sites had hypoxia, which is really it's large. It's about 40% of all the estuaries. Oh, the last thing I should mention is up in the northern part in the Bothnian Sea and the Bothnian Bay, there's only a handful of red dots and 1 or 2 black dots, and they're all associated with paper mills. So for a long time, paper mills were allowed to put fibers directly into the coastal area, and those fibers would sit on the bottom and consume oxygen. And then you have persistent hypoxia, and those are estuaries that have hypoxia 365 days a year, mostly. They could have occasional saltwater inflows, and there's 8 of those systems around the Baltic. A seasonal actually has a very relatively low number, and we would have expected it to be much more prevalent. And the next highest one is episodic, which has 30% of the time there is hypoxia. It's interesting, and it tells you something about the mixing processes in these shallow estuaries that are experiencing hypoxia. The example of this is the Limfjorden. It's in the northern part of Denmark. And this was, the first plot is taken in early August 2009. In Iltsvin, it's Danish for hypoxia, it's the red mark. And so that's less than 2 milligrams per liter. And the blue is less than 4 milligrams per liter. And actually, it's the other way around. So the red is less than 2, and the blue is less than 4. And you can see an area in the middle, the Limfjorden, in early August. By mid-August, this had spread really tremendously throughout the estuary and further down towards Skive, as you can see, and on the western side of the Limfjorden as well. And if you go 2 weeks later, it's nearly gone except on the western side. And so if you look at the data and you plot the data, all the different data points, and you can determine which ones of them are mixed and which ones of them are stratified. So when the wind doesn't blow, the estuary is stratified, the surface water heats up and the oxygen is consumed in the bottom waters. Wind comes along, it mixes, it puts oxygen back in the water, it can stratify again and consume the oxygen. So in many of these coastal systems, you have this up and down picture of hypoxia. So what are the mechanisms governing oxygen consumption? They're the same mechanisms that you see every place. Stratification of the water column is extremely important, whether it's from temperature or in the Baltic Sea, especially the open waters, it's about salinity. And what it does is it stops oxygen renewal from surface waters or from the atmosphere. The second thing is you need organic material. You need organic material produced from phytoplankton. So the nutrients come in and grow algae, and they are regenerated and consume oxygen. But there are other things, temperature, and that was also talked about by Wenfei. And temperature increases the rate of respiration, it increases stratification, and it reduces the saturation of oxygen in the waters. But there are other two cases that I want to talk about that's important for the Baltic. And this is from the GO2NE Deep, the mid-central basin, and the Baltic proper. And you can see the isopleths showing 2 milligrams per liter. And it's interesting because that also coincides with the permanent halocline in the Baltic, which ranges between 70 and 90 meters. So above that, the system is oxygenated. But below that, you can have these periods of time, as it happened in the GO2NE Deep, where oxygen is consumed and it becomes very hypoxic again. But what happens, as shown by the colors, is it releases phosphorus. So the iron-bound oxides are that they're released when it becomes anoxic or hypoxic, and also sulfate reduction happens. And so it releases of phosphorus up into the water column. And this has a big effect because you end up with something called the vicious circle. So if we look at the red line, it's when you have anoxia or hypoxia, you bring phosphorus up into the water column, and it could be mixed up in the surface. In a system like the Baltic Sea, you'll cause these huge summer cyanobacteria blooms, and there'll be nitrogen fixation. So there'll be more nitrogen in the water, more production that then settles to the bottle. So that we are kind of locked in this vicious circle because the nutrients are being recycled. And so we actually need to wait till phosphorus is either washed out or buried in the sediments permanently. And the last thing is about changes with warming. These were 2 different periods in the Danish Straits, and you can see the increase in hypoxic area with warmer temperatures between 1998 and 2002. It really changes the system tremendously. Again, you have more stratification, you have more oxygen consumption, and oxygen is less saturated within the water. So what's the future of the Baltic? The model suggests if we reduce nutrients that the Baltic Sea will improve. I would admit that this model was done earlier on, and not all them, it was from an ensemble of models, and not all the models were geared towards modeling hypoxia. But the more recent models also show this improvement with nutrient reductions. And so the pathway to a healthier Baltic Sea is to continue to reduce nutrient loading. It's really the only long-term solution we have. So there's 4 different perspectives I want to leave you with, and the Baltic Sea is naturally prone to hypoxia, and there were 2 longer periods of hypoxia during the Holocene. Modern hypoxia is clearly driven by nutrient input. It's more intense and widespread than we see from the sediment record in the geological past. Saltwater inflows modulate hypoxia in the Baltic. When saltwater comes in, it has some oxygen associated with it. It provides some short-term relief, but it enhances stratification, and hypoxia also increases. And unfortunately, it's probably that seasonal hypoxia is going to become more frequent and expand in a warmer climate. And it shows that we really need more nutrient reductions. So thank you so much for listening.
Thank you, Dan, for this very nice overview of the Baltic Sea hypoxia. There are a lot of questions here. First question, a lot of questions concern the possible solutions. So you mentioned the nutrient reduction is really the long-term solution that you consider as best solution. But here there are other questions about solution that you have read maybe in the press, etc. And one of them concerned the possibility to inject oxygen. Do you think that it's a possible route to solve the Baltic Sea hypoxia problem?
So I didn't present any of this work today, but I've worked a lot on the so-called geoengineering. And so it's impossible to inject enough oxygen into the system. It requires on the order of 2 to 6 million tons of oxygen each year to keep it from going hypoxic. And there's no way through bubblers or any other way to inject oxygen into the system. There have been questions about mixing the Baltic Sea. There's been one small scale experiment and it has improved a coastal estuary, but there are many questions related to if you do it in the Baltic, what are the ecosystem consequences? We have a system that has been permanently stratified throughout its history. What happens to the biology, for example, if you destratify the system? So there are a lot of questions on whether this is practically possible or financially possible as well.
Yeah, and there was a similar question about solution. And here it's proposed to, what do you think about seaweed aquaculture and marine permaculture to restore the ecosystem? Can it play a role?
So there's been a lot of studies on mussel culture, and can mussel remove algae at a sufficient level to change the phosphorus? And the answer is basically no. The coastal zone is too small for mussel farms to really do this this job. One of the problems is mussels in the Baltic are the size of your thumbnail, and where you're used to in the North Sea, the size of mussels that you can eat. And so there's just not the biomass that's there to do this. And one of the things we talk a lot about, what we really need to do is we need to remove the nutrients before they get into the Baltic Sea. So either through sewage treatment or through agriculture or other ways to reduce the amount of nutrients going into the system.
Yeah, there is also a question about the importance of atmospheric input. Do you think it's important in the modulation of the hypoxia cycle, this atmospheric nutrient input? I guess that it's about the nutrients flux from the atmosphere, yes, compared to the rivers.
Yeah, it's certainly a factor. But right now the rivers are dominating the nutrient inputs, and the runoff from land, from agriculture, and from sewage treatment plants have a much bigger effect than the atmospheric deposition. I mean, the good news is we're burning less CO2, especially in Europe. And when we have less combustion processes, the amount of nitrogen input has declined significantly in the Baltic Sea because of this. And atmospheric dust really doesn't play a big role in this ecosystem either.
Okay, there are many questions, so I will forward them to you afterwards. Maybe one or two last question. There is one about in terms of nutrient loading, the phosphorus target is lower than the nitrogen target. Why is it like that? Is it because the system is more limited by phosphorus?
I think it's actually more limited by nitrogen currently. And so the concept is we need to reduce phosphorus more so that phosphorus can also be a limiting nutrient as well. So you see different limiting nutrients during different times in the system. So these targets were made with ecosystem models, and they did different scenarios, and they're trying to use the best targets available to reduce the amount of organic matter that will make it to the bottom and decompose and make for a better Baltic Sea.
Okay, and then you also show that the data sharing in the Baltic Sea is quite well organized. So there is a question about the data collection, and the question is it common for national government in the region to monitor the coastal oxygen hypoxia as part of the water quality monitoring parameters?
Absolutely. So the coastal monitoring programs in almost all the countries have been pretty substantial, but I think with the Water Framework Directive and the requirements to monitor the coastal zone, that has improved tremendously. And I must say that the countries are now sharing their information much more. I think there's a couple countries or groups that still you have to make special requests to get the material, but almost all the countries are putting all their data into open databases that everyone can access. So it's a big change from a decade ago when they were all holding on to their own data, and it's a really good thing.
Yeah, it's a very good example to follow. Yeah, that's very good. Thank you so much, Dan. Thank you so much for this very nice talk, and also thank you to all the participants for this many questions. That was very interesting. So unfortunately, we do not have time to take all of them, but there are many. So now I guess it's time to thank again our two speakers. They give very great talk. It was very nice. And to conclude, I would like to remind you that you can submit abstract for giving a talk. So here you can see on the slides that the address where you can submit your abstract. It's also available on the webinar announcements. So, or you can also approach member of the Goal group directly. So that's up to you. We are really looking for people to make presentations. So do not be shy and try to present your work. We'd be very happy. Then the story is continuing, and we are planning the next webinar on March 17. And so before that date, I just have to close the webinar and to wish you a good end of the day. So thank you so much, and bye-bye.
Thank you. Bye-bye. Thank you for listening.
Bye. Thank you so much.